Hello and welcome to Delve Deeper, the MASC's 2024 webinar series that plunges you into the deepest depths of marine science without the need to make a safety stop at the end. Today we are embarking on a planktonic journey as we explore the groundbreaking world of environmental DNA meta-barcoding in aquaculture pathogen detection. So without further ado, let's give a warm welcome to our esteemed speaker. Their research focuses on plankton and environmental DNA, aiming to understand the coexistence of multiple species in ecosystems. Inspired by Hutchinson's paradox of the plankton, they explore phytoplankton dynamics as a model system to investigate and address ecological questions at local and regional scales. Their research interests also include the impact of anthropogenic activities on coastal communities and the impact of harmful algae blooms on coastal seaweed and finfish aquaculture. Speaking today on the potential of eDNA as a plankton monitoring tool for coastal waters and aquaculture, please welcome Dr. Sofie Spatharis.
Thank you very much. Thanks for the invitation, Phil. It's really great to be talking to a marine audience for a change. Phil gave a nice introduction, and thank you. So just to give you a bit of a history about my own research interest, using a more visual manner. So I like to work on theoretical questions that have to do with how species coexist using plankton as a system, and looking into how resource fluctuations might affect species coexistence. But I also like to ask questions that have to do with meta-community ecology, so different water patches connecting and how dispersal processes can affect phytoplankton assembly. But I also like to work with marine pollution questions, and more recently, since I moved to Glasgow, ask questions about aquaculture. We also do a lot of lab work regarding microalgae biotech. This is more an ongoing project regarding bioreactors, but my other more phytoplankton community research has this overarching theme that I really like to work with multiple species, which of course adds a further complexity than looking only on populations, but I really find that intriguing. So what I'm going to present you today are questions that relate to my meta-community work, but also aquaculture. So they fall in the interface, and they both have the coastal system as the common denominator. I really like to work on coastal systems to address such questions. Well, one thing is that I was so feeling seasick in a big oceanographic boat, so there was no chance for me with that part of research career. So I decided to play it more coastal with zodiacs as well as lab. But also because the coastal system presents some really nice features to investigate this question. So for instance, we have these interactions with the land with respect to nutrient concentrations and loading, interactions with the sediment, of course. We have interactions with the open sea, because I like to work on enclosed gulfs that have some communication, albeit limited, with the open sea. And then we also have increasing aquaculture, which of course interacts with the coastal ecosystem both ways. So we have the coastal system affecting the aquaculture and vice versa. All of these features are, of course, becoming more affected due to climate change. So we might have hydraulic changes in the water levels. We have increasing pollution because most of the human population tends to aggregate in coastal areas. And we also have a lot of infrastructure changes that again might affect hydraulic connectivity. So the group that I am focusing on is plankton, and luckily, due to Attenborough's Our Planet plankton focus recently. We have received more attention finally. But plankton has been tantalizing researchers in ecology since the 1960s and even more back. But it was mostly by Hutchinson's work, where he basically asked, why do we have so many species coexisting in just a drop of water? So that was called the paradox, and many studies have tried to resolve it ever since. But this is also showing what a great system is plankton to resolve biodiversity questions. So in today's talk, I really wanted to focus both on a more fundamental question that we were focusing on with the EU project back in my previous workplace in the University of the Aegean in Greece. But also a more applied project that has to do with plankton monitoring as an early warning system for aquaculture pathogens. So the first more ecological question has to do with dispersal, the role of dispersal over the environment in governing phytoplankton assembly. And the other one is the more applied, related to plankton. But for both of these questions, we have used morphological plankton identification as well as metabarcoding to look into how they can actually help us address these questions. So this is the common theme between these two quite different questions. And the tools are a classic microscopy that we are using to identify phytoplankton species and zooplankton. Whereas more recently, we have more tools in our basket, like the community metabarcoding, as well as eDNA metabarcoding. So we are increasingly concerned that microscopy might not do the job so well, because it is time consuming. We need people to know their taxonomy, but we are also not able to process multiple samples at the same time. On the other hand, the metabarcoding approaches, they are great. We start to have decreasing costs. We have increasing expertise. However, how sure are we that it can actually reflect what is there? So these are the more methodological questions I will be trying to address during this talk. So let me take you first on this first case study that we did in Greece, and this is to address the more ecological question of whether phytoplankton assembly, so the different species that make up phytoplankton, are more governed by neutral or niche processes. By neutral processes, I mean processes that have to do more with is a species there because it has been brought by, let's say, currents, or is it there because the environment has selected for it, has favored the growth? And to address this question, we used I don't know if you will think this is a broader scale, but we used the Aegean Sea in Greece, and we monitored 9 different gulfs during a very brief period of time, during a month in July 2014. We had 5 stations randomly selected within its gulf, and we had 3 different depths. So surface 1 meter, Secchi depth, and then 2 times the Secchi depth. And at these stations, we measured relevant environmental variables like dissolved inorganic nitrogen and phosphorus, temperature, salinity, but also we took samples for the plankton, which we identified under the microscope, and we also carried out molecular analysis using the 18S primers and Illumina sequencing. So what we found is that microscopy revealed much less diversity, well, not much less, but definitely less species than OTU that were assigned to species via blast using the Illumina approach. The species that we missed because of the microscopic analysis were mostly very small species that are not easy to identify in a microscope, and mostly of the classes Chlorophyta and Ochrophyta. So mostly we identified diatoms and dinoflagellates. whereas, as you can see, the Illumina is able to identify much more taxonomic groups. Can I just say here that we were only focused on the autotrophic, this is a mistake, autotrophic and mixotrophic microalgae. Then, again, as a very brief comparison between the two approaches here, we see that if we do a MDS approach with a microscopy and the molecular data, we see that our samples were much more different to each other based on their species abundance composition, so the actual identities of the species. Whereas the Illumina, they're all clustered together, which means that they don't really have a discriminating power, these data. Although, and actually this might be even the reason, we see that the frequency of samples that they had a specific number of species, we had much more samples in the Illumina approach that had many species on average inside than the morphological approach. So, with regards to the environment, our samples were quite different with respect to salinity and temperature, slightly less different with respect to nutrients, but we do have differences in the environment. And actually, what we are trying to do here is use this approach, principal coordinates of neighbor matrices, in order to understand whether the community assembly, so the differences in our samples based on their species abundance composition, how can this be partitioned to the component that is explained by environmental variables, so the profile of environmental conditions within its gulf, and to this spatial component that might reflect other things that we didn't monitor, and might give a hint about what other processes might be happening. So our hypotheses were if patterns in plankton composition are explained by environmental variables, which would reflect more a niche theory, or whether the patterns in plankton composition are explained by other spatial structures, and whether these align with any thermohaline circulation patterns in the Aegean, which would indicate dispersal patterns and therefore more neutral processes. So what we found is that the morphological data were reflecting more other hidden spatial structures. So this is the variation explained, whereas the molecular data were more sensitive to environmental cues. So basically, we get a very opposing signal by either using one or the other kind of data. And indeed, the morphological data seem to reflect more two spatial structures, which I will explain in a second. So PCNM1 and 2, and salinity, whereas the molecular were a bit more sensitive for dissolving organic nitrogen and temperature. So if we focus now on the morphological data, stations that were on the east-west gradient seem to actually be very separate to each other. And that is either using species abundance data that we identified in the microscope or just presence-absence data in the microscope. So this was reflected by these two PCNM variables, which the first one reflects this east to west gradient, and this that reflects a further gradient on the north east, that again can be explained by what we found out afterwards, is the thermohaline circulation in the Aegean Sea. So what happens here is something quite interesting, and it has been already published in the past. So we have the very cold and low salinity water coming from the Black Sea here in the north, and that water tends to flow along the north, and then is it east or west coast here? Eastern coast of the Greece, of the Aegean, whereas the water that is more saline and high temperature coming from the Mediterranean and the Levantine tends to flow upwards along the Asia Minor Turkish coast. So this actual pattern with a small separation here where we have the mixing of the masses seem to actually be explaining a lot of this variation, indicating that at least based on the morphological species abundance composition, it is more dispersal processes that are more important. So marine compartmentalization instead of environmental variables. However, although the molecular data were not able to actually depict the species abundance composition via an MDS, they were able to reflect this pattern, and even better than the morphological, when using the diversity of OTUs. And we can see that again, the stations that were on the east all had much higher species diversity than the ones that were coming from, had the influence of the Black Sea water that were on the east. So that was quite an interesting finding, because although they show the same pattern coming from two different directions, we do have an agreement and a complementary contribution of these two approaches to address an ecological question. So with respect, though, to just focusing on the metabarcoding data, and again on this same data set of the Aegean, we had this newer study by Alexandra, which showed the contribution, an increased contribution of parasites. So now we focus not only on autotrophic and mixotrophic species, but the whole community. And she actually showed very recently that if we take the whole plankton community of these samples, we have a surprising increased contribution of chytrids and syndyniales, which are parasitic organisms inside dinoflagellate cells. So they must play a very important ecological role in these systems. And we were able to understand this contribution by analyzing this metabarcoding data. And we even see how these contribution can change with depth. So we have we see more autotrophic organisms at 1 meter depth, and this decreases with depth, which is quite intuitive, whereas parasites were present across depths. And parasites were also contributing quite strongly in trophic networks. If we do a network analysis using trophic groups, but again, the networks might break as we go deeper. However, they were contributing quite significantly in interactions with other trophic groups. So that was, these were some insights regarding the more ecological studies that we have done. And to summarize from this part, microscopy data were better in reflecting differences and discriminating more with respect to taxonomic composition, so actual identities of species separating stations, whereas molecular data were more exhaustive and sensitive in detecting species, but they were able to reflect changes with respect to biodiversity. But they are actually quite good when we are talking about the whole community and not only focusing on the more autotrophic part of the assemblage, but when we are looking more into the more complex community, of course, molecular data are able to show us much more insights, and also the complex trophic networks that microscopy data are, of course, unable to do that. So now I'm going to pass from Greece to Glasgow, to Scotland, and I'm going to talk to you about our more applied coastal work that we do with eDNA metabarcoding. And this is in collaboration with Martin, my colleague here in Glasgow, as well as Mary, who was a postdoc working on the Salmon Health Project. So we have two concurrent projects going on. The one is we work with different salmon producers here in Scotland to understand what is driving the pathology of fish related to gill health. And we have another project that has to do with seaweed biofouling and how can, and in both cases, basically, how can we predict which plankton-borne vectors will then settle into either the gills or the seaweed and cause problems in the production and animal welfare, of course, in the case of salmon. So I'm not going to talk about the seaweed biofouling project today because this is ongoing and we are still trying to put the eDNA data together, so I could not offer any comparisons with that. But for the salmon project, we have already some published work, and this is what I'm going to be presenting. So the problem here is what is called proliferative gill disease, or PGD, as I will refer to it from now on. And this can be caused by different plankton-borne organisms like algae, but also some zooplankton species. More often, they are gelatinous zooplankton, but not always. And these can cause irritations to the gills, and they are very bad for fish welfare. They can cause enormous financial losses annually of millions. And there's not much we can do about them, especially when we cannot know when they're going to be happening. So that is very crucial. So the problem here is that with increasing temperatures, as we see also from the heat waves this summer, these can get much worse. So the problems are only increasing, unfortunately, with SST rises. So solutions need to be found to make sure that this sector has a future. In these approaches, we used environmental DNA. So basically, we filtered water, and we used this. Well, actually, it was Martin that adapted this pumping machine that he bought from Amazon, and it works very well because then we put our Sterivex filters. We filter 500 milliliters of water. And actually, it was not us, it was our actual aquaculture colleagues that did that. So it had to be relatively straightforward, and it was. And then these filters were sent to us in Glasgow for further analysis. We used CO1 primers for amplification, and we used Illumina for sequencing. So we know that this eDNA approach can work in principle. We had investigated in the past during a laboratory experiment using specific species of zooplankton and phytoplankton, including sea lice, but also some phytoplankton species like Pseudonychia and Primnisium parvum, which are harmful algal bloom species. And we actually investigated the sensitivity of two sequencing approaches, both Ion Torrent as well as Illumina, and we actually saw that they worked pretty well, most of the cases, even able to recover abundances of the different species in the cultures. So we took this and we tried to upscale it. I don't know if that's the right word, but to see if it works in the field by trying to see if by using two different sites for comparison, aquaculture sites, and exposed and a sheltered one with respect to wave action. And we obtained samples from these two aquaculture sites on a daily basis for plankton eDNA, but we also had microscopy for phytoplankton and zooplankton. And then we also had accompanying information on fish gill health, mortality, as well as temperature and oxygen. So what we try to do is then analyze this data to see whether we can find predictors of the PGD, so the pathology of the gills, using both morphological and eDNA plankton data. We did have some hit lists of potential suspects from the literature that are known to cause these problems on fish. So we were, really targeted in seeing these species, but we actually monitored everything in our samples with both microscopy and eDNA. This is a general overview of the pathology. So we had increased mortalities during usually the summer months, and then also more increased scores of proliferative gill disease. Plankton and zooplankton, phytoplankton and zooplankton presented different peaks, but yes, especially in the sheltered side, they were more pronounced during the summertime as well. So the eDNA did a very good job. This is the coverage, the temporal resolution of our data. So it did quite a good job in retrieving different phyla. However, as you see here with the red, many of the OTUs were unassigned to a specific species or genus, or to anything at all. So this unassigned was not even assigned to a different phylum. And we can generally see that we have a lot of amoebae and a lot of diatoms being detected by this approach. And generally, we did manage to see quite a lot of our target hit species by the eDNA, and also recorded at a species level. However, in general, the read depth was quite low. And we can also see that the temporal autocorrelation of these are the main predictors based on the eDNA approach, and this is based on the morphological approach. You can see that during time in the two different sites, I think this is the sheltered and this is the unsheltered, we had much more temporal autocorrelation in our microscopy data than we had on our eDNA. And not having temporal autocorrelation is not so great for being able to do analysis for prediction. We did find with both approaches some old culprits of fish health, such as Obelia, Pseudonychia, Prorocentrum, Hetocerus with the diatom, and Lycia blondina. So all these had been reported in the past, but we also had some new surprises that were previously not reported on the literature. And this was this interesting Appendicularia species, marine tunicate, Oikoplevra dioica, this jellyfish, Rathkia octopunctata, Doliolum, again, gelatinous zooplankton. We also had Rhizosolenia and Ceratium being indicated by our analysis as closely associated to incidence of mortality or PGD. So what is interesting is that some of these species were not found by eDNA. So Oikoplevra, where there was quite a strong signal by our microscopy data as a predictor of gill health, was not detected by eDNA metabarcoding. And that was because it was not even existing in the gene bank. So there was no reference sequence. This is a very interesting species, gaining more increasing attention. This is a mucus bag that it excretes from its stomach. It captures different bacteria, detritus, and then brings it back into its mouth. And it might be that this mucus might be something that irritates gills of the fish when it is being excreted, and these are in high abundance in the water column. Another marine dinoflagellate, Ceratium tripos, was also not detected by the eDNA. And here I will try to say this properly. Martin thinks it's because the primer was not able to bind properly because of not having a SNP in the primer sequence. I'm not really sure I'm saying this right. Forgive me if I'm not, but it was because to be honest, in our previous study using 18S primers in the Aegean, we were able to recover Teratium species. So it was a bit of a mystery and unfortunate that although these species were very abundant in our samples throughout the period, but with very distinguishable peaks, we were not able to detect them at all in the eDNA metabarcoding. And this is my last table, but let me stay on this a little bit because it actually compares the two approaches using our aquaculture data. So what we see in this table are only taxa that have been identified at this level by both approaches, so eDNA and microscopy. What we see in this first column is the percentage of samples that this approach, that microscopy managed to identify these samples. And we see that, sorry, this is in how many of our samples the two approaches found this taxon at the same time. So we see that this varies between, I don't know, 40 to quite high numbers, 80, but we actually have, in many cases, that only in 50% of the samples, the two approaches identified this taxon at the same time, which is not great. And if we see in the second column, it is the number of times that the species was recognized by microscopy over eDNA. So the higher the number, it means that microscopy was more successful, whereas the lower than 0, it means that it was eDNA that was, or lower than 1, it was eDNA that was more successful. So again, we see a mixed bag here, that it is not always the one or the other that was more successful. And here we attempted to do some correspondence of the actual abundances. So we took the abundances of a specific taxon as we identified it by microscopy, and we also took the reads. We plotted them and we found the regression line. And as you can see again, for where I don't present it, it was because the relationship was not significant. There was pure noise. In some few cases, we do see a relationship which might show for some, at least taxa, a potential for optimizing an abundance approach using eDNA. The best relationship was with Chiripedia, surprisingly, which are the barnacles. And I'm not really sure if that was because of eDNA that was shed by barnacles that were already settled as an adult in benthic substrates, or because of actual planktonic larval forms. So yes, that's me. These are the people that I have been working with, both in the Aegean University as well as here. A big thanks to my colleague Martin, who has been driving a lot of the salmon project, as well as our collaborators from Norway, and Mary, who was the main postdoc working on this. And yeah, I can stop sharing. And I don't know if you have any questions.
Thank you so much for that. This is such an interesting area. Like, I feel as if I don't know enough about the uses of eDNA. So this has been like a genuinely really exciting talk for me today. We are going to move into the discussion / Q&A part of the webinar now. There are multiple ways that you can ask a question if you have one yourself. You can use the Q&A section, the comments section, or you can just simply raise your hand and unmute yourself, turn your camera on if you want to as well. I will remind people we are still recording, so if you don't want to be featured on YouTube, totally get it. So you don't have to turn your camera on or your mic if you don't want to. But again, you can use the Q&A section. Speaking of which, let me open that just in case there are questions coming in. So I'm going to kick things off. So I think one kind of question that came to mind throughout the talk was, so how accurate is this as a method to sort of um, what have I written here? As predicting potential conditions. So considering an early warning sign, if I was to set up a new aquaculture farm, how useful is this technique in giving me information about how my farm might do?
I think we are at very early stages with respect to eDNA. I think we still need to make sure that all the species that we see with our eyes in the microscope, and we know they're there, can be detected to begin with. So we obviously need appropriate primers, even combinations of primers, and we need to be able to curate the databases with species that are not yet there and that we know they should be there in order to be able to detect them when we do this kind of analysis. So I think that it is an ongoing work. We are not yet there, but obviously it has a big potential, and we should not give up because it does recover so much more than microscopy, because obviously it's with microscopy, we are error prone because of our own personal expertise. There are time constraints because these samples take a lot of time to investigate, and aquaculture needs fast decisions sometimes. Or if this data feed into models and machine learning that will give us an early warning, like you need to apply this mitigation right now, this needs to be very streamlined and fast. So yes, there is still work, but we should still be doing this work. That's my point.
So actually, one of my first ever jobs in research was identifying species under a microscope for water quality studies. And I remember myself having so many problems identifying, like I would see something and be like, I don't actually know what this is, and I can't really see it on this list. And that kind of brings up another interesting question: is how um susceptible, well, how sturdy is the wrong word, but in terms of things that can affect results of eDNA, how susceptible is it to these sort of things? So considering things like storms, other forms of sort of sound and general pollution, like can this affect the results of eDNA, or is it a subject that is a lot more accurate regarding of the conditions?
And so, for instance, in this analysis that we did in Greece with the different gulfs, we had some problems with a specific gulf, Thermaikos, because not Maliakos, because that there happened to be a diatom bloom at that period. So it messed with our molecular analysis, and we weren't able to have data from that gulf on for the molecular data. And we think that it might have been biased by these excreted polysaccharides from diatoms during this bloom, because there was a kind the water was cloudy, and we think that it might have really interfered with the molecular analysis, but not with the microscopy analysis. So there are different things that can go wrong. And yes, testing the sensitivity of the method under different conditions, different temperatures as well, is very important, different salinities, I guess. Uh, Although I don't, yeah. So, but yes, definitely. But it should be an ongoing goal to try to improve it, because it definitely has potential, as we saw also from the parasite work, how much it unveiled with respect to the whole community and different trophic groups that we are not so, it's really hard. These microscopic taxa that are very difficult to identify with a microscope.
Yeah, as I said, my one of my first jobs, and I just remember I didn't last long in the job because of how difficult it generally was. Um, I am just cautious of time, so I'm just going to ask: does anyone in the room have any questions that they would like to ask? Up, we've got one hand up. Oh, Alexandra.
Hi, yeah, hello, Sophie.
Hi, Alexandra.
Hi. I just wanted to query you about your choice of primer for. Did you say that you used 18S in the Greek Gulf study?
In the Greek seas, yes, we did.
And then CO1. Was
yes.
Yeah. So yes, so just querying.
Why is the question?
Yeah.
Well, I will try to answer this, but I'm not sure I will do a good job. I think that the reason our molecular experts in the Aegean project wanted to use 18S because we were focusing more on marine protists, and they thought that this will recover more that kind of diversity. Whereas we used the CO1 in this approach with the aquaculture because we were also focusing on the zooplankton component as well as the phytoplankton. We thought that it might work better in capturing both. But from what I feel, it kind of underestimated the protist and more microalgae diversity.
Okay.
Yeah.
Thank you.
Thanks.
Thank you very much, Alexandra. One last one last shout out if someone does have a question, because we've got a few minutes left and I don't want to take up all the question time. So yeah, you can use the raise hand function, or you can just write it in the Q&A or the comments. I'm trying to keep my eye on all three sections, which is why they're darting back and forth. So one question I kind of, even before your talk, kind of popped into my mind straight away, and I'm apologize if I think you've covered parts of this during your talk, but what are, in your opinion, the main challenges of kind of implementing this on an industry level? Because obviously this is something that would be very valuable to, well, any aquaculture farm.
Yes, so the ultimate goal in such an approach is to really find which are the important predictors for fish disease, and then try to find more targeted primers for those, create some sort of a paper-based device that aquaculture colleagues can very easily even dip in the water on a daily basis and see if we have an increase of this kind of pathogen in the water at that time. So ideally, that's the ultimate goal. But of course, we have some way to go until we reach that stage. But that is something that would work quite well in the future of aquaculture. Until then, some intermediate solutions of course, having some sort of more, how do you say, an actual device in place of the aquaculture. It depends on what kind of aquaculture we're talking about as well, because salmon industry tends to be more rich than seaweed, for instance. So the amount of support that is needed with respect to collaboration with the academia is also varies. But yes, in the beginning, it will need to be collaboration with either. Yeah, it still needs a lot of work. It's so far away that I cannot tell you yet how it will work now.
I mean, it's very exciting, and I keep seeing, as say, eDNA is. I know it's been around for a while, but it's still a concept to me that's quite new. So I feel like I'm learning things about it all the time. I mean, that question as well kind of answered my next question, which was on scalability, especially in places where there are sort of limited resources, or where cost is an issue. But the idea of something that is as simple like a lab on a chip type solution sounds incredible.
Yeah.
I guess my final question that I have, and again, this is we are almost at the end, so if you do have a question, this is your time to ask it. But so we've spoken a lot about how eDNA can be used for pathogen detection, how it can be used for specifically in things like you mentioned PGD and other parasites. Are there any other applications that are potentially useful to the aquaculture industry that this sort of that eDNA monitoring can be that it can be used for, or is this kind of the main topical?
So in my opinion, I today presented to you how the environment might be a threat for the aquaculture, but it can also be the other way around. It can also be the aquaculture presenting some sort of danger by by exacerbating by nutrient release or concentration of these organisms via the nets, because we create a substrate by creating aquaculture. Even seaweed and salmon and all forms of aquaculture creates a substrate. So that might act as a form of release of these organisms in the environment. So one way is the vice versa, to monitor changes in water quality around aquaculture by using appropriate controls as well to test the environmental impacts.
Yeah. So that almost sounds, because I do a lot of wild swimming, and there's been several times we've been at a loch and we're wondering, is this a loch we should be swimming in? Is this one we shouldn't be swimming in? And we're always a little bit dubious, especially after heavy rainfall. So something like that, that's a really interesting. I really need to start reading more into eDNA, because it sounds like the possibilities, like it's got a very exciting future ahead of it, which is always, yeah, I think watch this space for definite. I am going to bring the webinar to the end. So I'd like to thank our speaker. Thank you so much, Sofie, for doing this today. It's been a really exciting talk, and yeah, thank you so much for being here. Thank you to everyone else who has joined us today. As I mentioned at the start, we will be uploading this talk to YouTube, and I will be putting it in the newsletter at the end of the week when you can go and view it if you want to rewatch it or share it with any colleagues who weren't able to make it today. So with that, I'm going to end the recording, and just once again, thank you very much.
Thank you very much for inviting me, Phil, and everyone, thanks for joining.